Basics of Medical Biochemistry. First edition, 2023. Chapter 18. Transcription and Translation. Part 1 of 2. By Professor Dr. Muhammad Jawad. MBBS, Tao, M.Phil Philosophy, Ph.D, Dip M.E. Head of the Biochemistry Department-Fazaia Ruth Pfau Medical College Air University, PAF Base Faisal-Karachi-Pakistan. Ex-Professor of Biochemistry, Qassim University Kingdom of Saudi Arabia. Ex-Professor, Head of the Biochemistry Department and Vice Principal. Liaquat College of Medicine and Dentistry and Fatima Jinnah Dental College, Karachi, Pakistan. Chapter 18, Part 1 of 2. Transcription and Translation. 18.1 Introduction. The flow of information in the cell starts with DNA, which replicates to form more DNA. Information is then transcribed into RNA, and then it is translated into protein. The proteins do most of the work in the cell. Transcription and translation are the two major steps in the expression of a gene. Figure 18.1 Relationship between replication, transcription, and translation, central dogma of molecular biology. In eukaryotes, the processes of transcription and translation are distinct both spatially and in time. Transcription of DNA into mRNA occurs in the nucleus. Once mRNA is completely transcribed, it is transported to the cytoplasm. Translation of mRNA into polypeptides occurs on polysomes, complex of ribosomes, in the cytoplasm. At the end of translation, the mRNA is degraded. In prokaryotes, which have no nucleus, both transcription and translation occur simultaneously. The newly formed mRNA molecule begins to be translated even before its transcription from DNA is complete. In human cells, there are 23 pairs of chromosomes. They contain so much coded information in DNA which is equal to about 1,000 big textbooks. Another fact is that only about 2% of the total genome represents genetic material. 18.2 Transcription. It is the process of copying genetic information contained in DNA in the form of RNA by a multi-enzyme system. It is a highly selective process. It results in the formation of three kinds of RNA, i.e., mRNA, tRNA, and rRNA. The mRNA formed with a particular sequence of N bases is decided by genetic information on DNA. By the combined work of mRNA, tRNA, and rRNA, a specific protein is synthesized, translation, within the cell. The complete set of transcripts expressed by the genome is called transcriptome. After synthesis, mRNA undergoes many modifications to become an active and functional molecule. 18.2.1 Transcription in prokaryotes. Following are the requirements for transcription in prokaryotes. RNA polymerase. 2 DNA molecule. 3 nucleoside triphosphates, i.e., ADP, GTP, CTP, and UTP. 4 Mg2+ ions and many regulatory proteins. Steps of transcription in prokaryotes. The process of synthesis of RNA, transcription, in prokaryotes, example in E. coli, can be described in the following steps. 1. Binding of RNA polymerase to the DNA double helix. 2. Unwinding of the DNA helix. 3. Initiation of RNA synthesis. 4. Elongation of RNA chain. 5. Termination of RNA synthesis. 1. Binding of RNA polymerase to the DNA double helix. The process of RNA synthesis begins with the binding of the RNA polymerase molecule to the template strand of DNA at the promoter site. The stretch of DNA that is transcribed into an RNA molecule is called a transcription unit. RNA polymerase. Prokaryotic RNA polymerase is a zinc-containing multi-enzyme system that can recognize the promoter region at the beginning of the length of DNA that is to be transcribed. It needs Mg2+ as cofactor. It can be made a complementary copy of the DNA template and finally can recognize the terminator region. In prokaryotic cells, all three RNA classes are synthesized by a single RNA polymerase. The prokaryotic RNA polymerase has the following parts. I-core enzyme. It is made up of 5 polypeptide subunits, 2 alpha, 1 beta, and 1 omega subunit. It is responsible for 5-3 RNA polymerase activity. The functions of different subunits are: alpha and subunits, they detect the DNA region which is to be transcribed. Beta subunit, it performs polymerization. Beta subunit, it performs the unwinding of DNA. 2 holoenzyme, the sigma subunit with core enzyme make up the holoenzyme. It scans the DNA and enables the RNA polymerase to recognize the promoter region on DNA. Figure 18.2 Structure of prokaryotic RNA polymerase. A transcription unit extends from the promoter to the terminator region, and the product of a process of transcription by RNA polymerase is termed primary transcript. RNA is synthesized from its 5' to 3' antiparallel to its DNA template. The function of unit in vivo is not clear. Promoter region, it is the region of nucleotide sequence at the beginning of a stretch of DNA that is to be transcribed. Prokaryotic promoter region. I-TGACA, -35 sequence. It is a consensus sequence of 5 TGACA3 situated 35 nucleotides to the left, upstream, of the initial base of the DNA strand used to code for RNA synthesis. It is the initial site of attachment of RNA polymerase. 2 Pfau box, -10 sequence. It is a stretch of 6 or 7 nucleotides, 5-8-3, situated about 10 nucleotides to the left of the initial base of DNA strand used to code for RNA synthesis. It is also known as the TATA box. It lacks GC base pair and has a low melting point. It guides the RNA polymerase for its attachment to the promoter region. It allows easy melting and formation of transcription bubbles. Figure 18.3 Structure of prokaryotic promoter region. The binding of RNA polymerase with the promoter region of DNA is carried out by the sigma factor. The binding causes a decrease in the affinity of RNA polymerase to combine with other regions of DNA. 2 Unwinding of DNA helix. The unwinding of DNA molecules takes place from the promoter region by RNA polymerase. One of the types of RNA polymerase, by its unwind-DOS activity, can open up about 15 to 17 base pairs of the template strand. By unwinding, the template become accessible for incoming ribonucleoside triphosphates. It is a transition stage from the closed promoter complex to the open promoter complex. Supercoils in DNA produced during unwinding are relieved by DNA topoisomerase. 3 Initiation of RNA synthesis. The initiation starts with the attachment of RNA polymerase to the -35 sequence of the promoter region, followed by the local unwinding of DNA in the region of the Pfau box. This activity results in the formation of the open complex. The initiation of RNA synthesis takes place at the 5 ends of adenine or guanine triphosphate residue, and primer is not needed. Nucleoside triphosphate is used and pyrophosphate is released each time during the addition of a nucleotide. As a result, a short stretch of 5 to 10 bases containing RNA is produced in the transcription bubble. Now the RNA polymerase moves forward to start the next step of transcription, i.e., elongation. The event of leaving the promoter region by RNA polymerase is called promoter clearance. Sense or coding, non-template, strand. It is the strand of DNA that is used for coding. It has the same sequence of N bases as the RNA transcribed, except for thymine and sugar. Antisense or non-coding, template, strand. It is the RNA-forming strand of DNA. It is a non-coding strand. One strand of DNA acts as coding for some RNA and non-coding for other RNAs. For elongation of RNA chain, the direction of elongation is 5 to 3 antiparallel to the DNA template. The localized melted region of DNA with RNA polymerase and nascent RNA is termed a transcription bubble. As a result of elongation, a short stretch of DNA-RNA hybrid helix is formed. But RNA from this hybrid is separated shortly after its synthesis. The speed of formation of RNA is 50 to 100 nucleotides per second. The overall process of elongation can be expressed by the following reaction. Nucleoside monophosphate, N plus nucleoside triphosphate, nucleoside monophosphate, N plus 1 plus PPI. Figure 18.4 Step of elongation in prokaryotic transcription. After the addition of about 10 nucleotides, the sigma subunit is removed from the hollow enzyme and combined with the next core enzyme and repeats the process. Unlike DNA polymerase, RNA polymerase cannot perform proofreading. However, in case of incorrect addition of ribonucleotide, the RNA polymerase stops, moves back, removes the defective transcript and restarts the process. The proofreading activity of RNA polymerase is supported by a protein bridge, which controls the entry of ribonucleotide to the active site. The base sequence in RNA is complementary to the corresponding part of the template strand of DNA, with the following specification. U is found despite T. These bases are stabilized by hydrogen bonds. There are 3 hydrogen bonds found between G and C, whereas only 2 hydrogen bonds exist between T and A or A and U. Figure 18.5 Sequence of N bases in DNA, coding, strand, DNA template strand and nascent RNA. 5. Termination of RNA synthesis. The termination of the process of transcription is signaled by a sequence in the template strand of the DNA molecule. Termination can occur in the following 2 ways. Rho dependent termination. It is carried out by the involvement of an additional protein called the Rho factor, which has an RNA-dependent ATPase activity. It also possesses helicase activity. Rho protein binds to a specific area, which is rich in cytosine bases near the 5' end of nascent RNA. When RNA polymerase reaches the termination sequence, termination factors contribute to separating RNA polymerase and elongating factors from the template and release of the transcript with Rho protein. 2. Rho independent termination. It is also known as intrinsic termination. It is carried out by RNA polymerase, sometimes only, by recognizing the terminator region on DNA. It is achieved through formation of the stable hairpin turn, which slows down the progress of RNA polymerase. The hairpin turn of the RNA is complementary to a region of the DNA template near the terminator region, palindromes. Near the base of the stem of the hairpin turn, G and C are richly found. This stabilizes the structure of the hairpin. Following the hairpin turn there is a string of Us. The bonding between U and A is weak, which helps the separation of newly synthesized RNA from a DNA template. Figure 18.6 Rho independent termination in prokaryotic transcription. Figure 18.7 Steps of prokaryotic transcription. Anti-termination. Some genes' structure includes a single promoter for many adjacent genes. When RNA polymerase arrives at this site, transcription is terminated by the termination. Thus, the gene distal to this site cannot be transcribed. This problem is overcome by the anti-termination factors. They help the RNA polymerase to bypass the termination site and continue transcription through the distal gene. These anti-termination factors include NUS-A and NUS-B proteins. Figure 18.8 Role of anti-termination factor in transcription. Clinical aspect. Some antibiotics can block the process of transcription at various steps. For example, rifampicin or rifampin, an antituberculosis drug, inhibits the initiation by interfering with the beta subunit of prokaryotic RNA polymerase. Similarly, actinomycin D inhibits initiation and elongation by occupying DNA bases. It is used in tumor chemotherapy. 18.2.2 Transcription in eukaryotes. Although the main steps and mechanism of eukaryotic transcription are the same as that of prokaryotic transcription, but it is more complicated and involves about 50 different types of regulatory proteins. The following are the requirements for transcription in eukaryotes. RNA polymerase. 2 DNA molecule. 3 general transcription factors, GTF. 4 specific transcription factors, STF. V nucleoside triphosphates i.e., ADP, GTP, CTP, and UTP. 6 Mg2+ ions and many other proteins. RNA polymerase. There are following 3 distinct classes of RNA polymerase. RNA polymerase I, responsible for rRNA synthesis, 28S, 18S and 5.8S. RNA polymerase II, synthesizes the mRNAs and certain small RNA like snRNA, snRNA, and mRNA. RNA polymerase III, synthesizes the tRNAs and 5s rRNA. Eukaryotic promoter region, for RNA polymerase II. ITATA or Hogness box, it is the stretch of nucleotide bases situated 25 bases upstream of the transcription site on the DNA molecule. In many genes, instead of the TATA box, different core prompter elements like initiator, INR, or downstream promoter element, DPE, are found. These sequences act as a binding site for many general transcription factors, GTF. They interact with one another as well as with RNA polymerase II. 2 CAAT box, it is situated about 70 to 80 nucleotides to the left of the initial base of the DNA strand used to code for mRNA. Figure 18.9 Structure of eukaryotic promoter region. General transcription factors, GTFs. These include many transcription factors like TFA, B, D, E, F, and HTF strands for transcription factors and 2 for the RNA polymerase II. The functions of GTFs are: identification of promoter region, placement of RNA polymerase II, formation of the pre-initiation complex, start of transcription at basal level. Specific transcription factors, STF, or transcription activators. These are special kinds of proteins having 2 binding domains. One is attached to regulatory elements on DNA and the other is a transcription activation domain that recruits GTF to the core promoter and many other coactivator proteins. Example HAT, histone acetyltransferase, enzyme, responsible for chromatin modification. Important STFs are CAAT box transcription factor, CTF, and specificity factor 1, SP1. Regulatory elements. These are consensus sequences situated upstream of the core promoter. These are arranged in the following 2 parts. Proximal regulatory elements. These are closed to core promoter elements, i.e., within approximately 200 nucleotides. They include a CAAT box and a GC box. STF like CTF binds to the CAAT box and SP1 binds to the GC box. 2 distal regulatory elements. These are situated far from core promoter elements. They include various enhancers. Steps of transcription in eukaryotes. The process of transcription, example formation of mRNA, in eukaryotes can be described in the following steps. 1. Binding of RNA polymerase II to the DNA molecule. 2. Unwinding of the DNA helix. 3. Initiation of mRNA synthesis. 4. Elongation of mRNA chain. 5. Termination of mRNA synthesis. 1. Binding of RNA polymerase II to the DNA molecule. The process of mRNA synthesis begins with the binding of RNA polymerase II to the template strand of DNA at the promoter site. RNA polymerase II cannot recognize the promoter region. The promoter region is recognized by TFA, which contains TATA binding protein, TBP, and TATA associated factors. It binds to the TATA box. Then TFA helps in binding RNA polymerase II to the promoter region. 2. Unwinding of DNA helix. The unwinding of DNA molecule takes place at the INR region of the core promoter element after binding of RNA polymerase II to the promoter region. During unwinding, DNA melting is caused by the helicase activity of TFA. This requires energy released from ADP hydrolysis. 3. Initiation of mRNA synthesis. The initiation starts with the attachment of RNA polymerase II to the promoter region, followed by the formation of the pre-initiation complex. The following factors are required for the formation of a pre-initiation complex. Their sequence of attachment to the promoter region is also mentioned below. TBP rightwardero Tfiad rightwardero Tfia rightwardero Tfiab rightwardero Tfiaf with RNA polymerase II rightwardero Tfia rightwardero Tfia. The aggregation of all these transcription factors at the promoter region results in the formation of a pre-initiation complex. Tfiad is the largest transcription factor, as consists of TBP and TAFs, TBP-associated factors. Its binding causes DNA to bend at 100 degrees. This bending allows and helps all transcription factors to work together cooperatively. TAFs may assist TBP in this binding process. Tfia possesses helicase activity, phosphorylation of the carboxyl terminal domain, CTD, of RNA polymerase II to activate it and nucleotide excision repair. Defects in the gene of Tfia may cause xeroderma pigmentosum. By the helicase activity of Tfia, local separation of DNA double helix takes place and the closed promoter complex becomes an open promoter complex. Now the initiation of mRNA synthesis takes place at the template strand of DNA in the direction of 5 to 3 ends. In human cells, TAFs are formed by 12 subunits. One of them is TAF 250, which has histone acetyl transfer activity that can relieve the binding between DNA and histone in the nucleosome. Promoter clearance. After the synthesis of a short stretch of mRNA, RNA polymerase II physically moves forward and leaves the promoter region. This is called promoter clearance. During promoter clearance, all transcription factors except Tfiaf are released. Figure 18.10 Step of initiation in eukaryotic transcription, TF, transcription factor, TBP, TATA binding protein. For elongation of mRNA chain, it is the addition of nucleotides to the 3 ends of RNA stretch formed on the DNA template during initiation. The added nucleotides are linked by 3 rightward arrow 5 phosphodiester linkage. The direction of elongation is 5 rightward arrow 3 antiparallel to the DNA template. This produces an mRNA molecule which is an exact copy of the coding strand but has uracil in place of thymine and sugar ribose in place of deoxyribose. RNA polymerase II uses nucleoside triphosphates, ADP, GTP, CTP, and UTP, to synthesize an RNA transcript. During RNA elongation, Tfiaf remains attached to the RNA polymerase II, but all of the other transcription factors have been removed from the pre-initiation complex. The CTD is the largest subunit of RNA polymerase II, and it is critical for elongation. It is activated by phosphorylation, which is catalyzed by Tfia. This domain is rich in proline and serine residues. RNA polymerase II also exhibits some proofreading activity as in prokaryotic transcription. 5. Termination of mRNA synthesis. The mechanism of termination in eukaryotic transcription is not well understood. There are 2 types of particular sequences on the newly synthesized RNA which act as termination signals. These are AAUAA and CA. To these termination signals, there occur attachment of special factors called cleavage and polyadenylation specific factor, CPSF, and cleavage stimulating factor, CSF, respectively. These special factors then recruit a special factor called the cleavage factor. This cleavage factor acts as an endonuclease and cleaves the mRNA from the DNA template. 18.2.3 Post-transcriptional modification. The newly synthesized RNA is called primary transcript. It is a linear copy of the transcriptional unit. Primary tRNA and rRNA of both prokaryotes and eukaryotes are post-transcriptionally modified by ribonuclease. Prokaryotic mRNA is generally identical to its primary transcript. Eukaryotic mRNA is extensively modified after the synthesis. Post-transcriptional modification in rRNA. Newly synthesized RNA of both prokaryotes and eukaryotes is called preribosomal RNA, 45s. The long preRNA of prokaryotes is cleaved into 23s, 16s and 5s ribosomal RNA by ribonuclease. Similarly, by the same enzyme, eukaryotic preRNA is cleaved into 28s, 18s and 5.8s ribosomal RNAs. The 5s ribosomal RNA of a eukaryote is synthesized by RNA polymerase III and is modified separately. Post-transcriptional modification in tRNA. Both eukaryotic and prokaryotic tRNAs are trimmed, shortened, after transcription. The addition of the CCA sequence at the 3-terminal takes place by nucleotidal transferase. Modification of specific bases also takes place to produce unusual bases like pseudouracil or N4-acetylcytosine, etc. Post-transcriptional modification in mRNA. In eukaryotes only the primary transcript of mRNA is called heterogeneous nuclear RNA, hnRNA. It is synthesized by RNA polymerase II. It undergoes the following modification. I5 capping. It is the first modification that takes place on hnRNA. It is the addition of 7-methylguanosine on the 5 ends of mRNA through 5-to-5-triphosphate linkage. This addition is carried out by a nuclear enzyme called guanine 7-methyltransferase. Methylation of guanine takes place in the cytosol by guanine 7-methyltransferase. S-adenosylmethionine is the source of a methyl group. By capping, mRNA is stabilized and is easily translated. This also protects the 5 ends from 5 rightward arrow 3 exonuclease and enhances the translation. 2. Addition of poly-A tail. Most of the eukaryotic mRNA has a chain of 40 to 200 adenine nucleotides at 3 ends. This poly-A tail is not the result of transcription from DNA. This addition is carried out by the nuclear enzyme poly-A polymerase. The addition of poly-A causes the stabilization of mRNA and helps its exit from the nucleus. It also protects the 3 ends from 3 rightward arrow 5 exonuclease. After entering into the cytosol, the poly-A tail of mRNA is gradually shortened. 3. Removal of introns. Intron is the region of RNA which do not code for protein synthesis. Exon is the actual coding region. For the maturation of mRNA, the removal of an intron is necessary. Splicing is the removal of introns from exons by the spliceosome. Spliceosome is a complex of 5 RNAs and many proteins. Its function is to splice out the non-coding introns from the primary mRNA transcript and stitches the exons back together into the mature mRNA transcript. Small nuclear RNAs, snRNAs, in association with special proteins called small nuclear ribonucleoproteins, snRNPs, facilitate the splicing of some exon segments by forming base pairs with each end of the introns. Figure 18.11 Post-transcriptional modification in mRNA, splicing. 18.2.4 Regulation of RNA transcription. Transcriptional control systems involve mechanisms that determine whether RNA polymerase can have a productive interaction with promoters. These mechanisms are reversible and can cause activation, positive, or repression, negative, of transcription. Positive regulation. In positive regulation, an activator, which is a protein, binds to the enhancer to facilitate and accelerate the binding of RNA polymerase to the promoter. Enhancers. These are the specific regions on DNA, not necessarily close to the genes they enhance transcription. When a DNA-bending protein, activator, binds to an enhancer, the shape of the DNA changes to allow the easy union of RNA polymerase to the promoter region. Negative regulation. In negative regulation, a repressor, which is a protein, binds to the silencer at the operator site and inhibits the binding of the RNA polymerase to the promoter. Silencers. These are specific DNA sequences capable of binding transcription regulation factors, called repressors. The repressor causes silencers to decrease the transcription of neighboring genes. Insulators. These are specific DNA sequences that prevent inappropriate interactions between adjacent chromatin domains. Insulators intervene in the contact between promoter regions with enhancers or silencers to avoid undue transcription and thus regulate gene expression. Figure 18.12 Effect of repressor on transcription. Table 18.1 Difference between prokaryotic and eukaryotic transcription. No. Prokaryotic transcription. Eukaryotic transcription. 1. Both transcription and translation occur simultaneously. Transcription and translation cannot occur simultaneously. 2. There is no definite phase for its occurrence. It occurs in the G and G2 phases of the cell cycle. 3. All three types of RNAs, mRNA, tRNA, and rRNA, are synthesized by a single RNA polymerase. The RNA polymerase I, II and III synthesize rRNA, mRNA, and tRNA respectively. 4. All steps of RNA synthesis, release and processes take place in the cytoplasm. All steps of RNA synthesis, release and processes take place in the nucleus. 5. RNA polymerase is a complex of 5 polypeptide chains. RNA polymerase is a complex of 10 to 15 polypeptide chains. 6. The transcriptional unit has one or more genes. The transcriptional unit has only one gene. Exercise questions. For MCQ 6 to 10, see Part 2 of this chapter. MCQ 1. Deficiency of which of the following RNAs is associated with heart diseases? hnRNA, mRNA, rRNA, snRNA, tRNA. MCQ 2. Which of the following RNAs has anticodon in its structure? mRNA, mRNA, rRNA, snRNA, tRNA. MCQ 3. Which of the following sequences of nitrogen base is the start codon? AUA, OG, UAA, UAG, UGA. MCQ 4. Which of the following enzymes is responsible to resolve the problem of supercoils formed before the replication fork during the replication of a DNA molecule? Helicase, ligase, polymerases, primase, topoisomerases. MCQ 5. During the translation process, the active site of the ribosome is facing the 5-ACC3 codon of messenger RNA. Which of the following anticodons would be on the corresponding tRNA? 3 August 5, 3 UG5, 5 AGU3, 5 AUC3, 5 August 3. End of Part 1 of 2 of Chapter 18. Thank you.